It's a great pleasure to have Jun coming to us from Johns Hopkins. She previously was at Princeton, and she's going to talk today about mechanical precipitation and bit separation in the cell nucleus by chromatin. Over to you, Jun.
Okay, thank you. Yeah, I want to first thank the organizers for the invitation here and for the opportunity to talk. I have been really enjoying my visit here. Yeah, so today I would like to talk about this recent work we had on mechanical fluctuation of phase separation in the cell nucleus by chromatin. Yeah, so since relevantly new, so let me briefly introduce me and my lab. I started at Johns Hopkins about 2 years ago. And so I'm jointly appointed in physics and biophysics departments there. I am a second-year PI, and I have 4 second-year graduate students and 1 second-year undergraduate student. So everyone is second year. Yeah, so our research, my main research focus is actually on biomolecular condensates, which is a topic of the workshop last week, and I really enjoyed that. Now today, since this week's topic is different, it's about chromatin organization and sulfate decision. So I pulled out this work, which is actually from my postdoc. I put out this work, and hopefully, which is on like the interplay between the condensate and chromatin, and hopefully it can still have some relevance to the topic of this work's seminar. Yeah, so I guess for this audience, I do not really need any introduction for the organization of the chromatin in the cell nucleus. Right? It is really like packed with hierarchically folded chromatin across multiple length scales. And I guess what let me just simply mention that, the cell nucleus is not just a home to chromosome, but it is also has a various of phase separated nuclear bodies. And so I'm particularly interested in the interplay between the chromatin and the phase separated condensates in the cell nucleus. Okay, so if you are not in the condensate field, many of you probably do actually, but if you are not, this is kind of a relatively new area, which starts about more than 10 years ago. And it has been discovered that a cell is a host of a multiplicity of biomolecular condensates. And these condensates, they are made of proteins or sometimes RNAs, DNAs, right, all kinds of the molecules. And they can form in the cell cytoplasm or in the nucleus or underneath the membrane. And they can respond to cells signaling, or they can also carry out crucial functions like ribosomal assembly or metabolism. So I guess these biological functions are how it gets biologists excited, and for physicists, we are more excited because of the underlying principle, which is phase separation, which is like really a physics tool. And understanding these phase separation of biomolecules is definitely goes beyond what currently the theory can explain. So there are way richer behaviors for the biomolecular condensate phase separation compared to like simple oil water type of phase separation. Yeah. And I'm now going to talk about theory. I guess I'll just say that, the simplest theory people use is called the Flory-Huggins model. All you need to know is that, from the statistical mechanics point of view, the purpose is always to minimize the free energy. And so say, because you want to minimize the free energy of the system at high temperature, then you could see all it matters is really the last term, which is T times the entropy, right? So entropy matters most. And entropy means randomness. So in this case, the molecules want to just run all over the place. And that corresponds to the well-mixed state. And so as you lower the temperature or as you increase the binding strength between the molecules, then at a certain point, the energy starts to matter. And at this point, the system will prefer to phase separate into two regimes, the dense phase and the dilute phase. In the dilute phase, you have high entropy, and in the dense phase, you have low energy, which makes you like both happy in the two phases. And if instead you have you are in a well-mixed state in that temperature, then you do not have this high entropy, and you also do not have this low energy. So that well-mixed state becomes unfavorable the mixed state. And that's why the system phase separate. Okay, so now let me draw your attention to the condensates in the nucleus. And many condensates, they reside in the cell nucleus and they interact with the chromatin. And in fact, the previous speaker is, was already mentioning, right? The nucleolars, they really exclude the chromatin. So you could see that like these nucleolars locations here, I mean, these nucleolars, when they are there, the chromatin, the first figure has holes, right? And this means that, these condensates, they push up the chromatin when they grow. And so as a result, there is a rich mechanical interplay between the chromatin and the phase separated condensates. And so then our guiding question is like, how does this chromatin impact the time evolution of the droplets, and as well as their steady state or equilibrium distribution. Okay, so now in order to understand how chromatin impacts the droplet evolution, maybe we should first go back to the simple case, right? We kind of go through this control experiment. And in this control experiment, it's really just we put the small droplet into the solution and watch in a simple liquid solution and watch how they evolve with time. And for such problem, physically, if we start with many small droplets, there are essentially two mechanisms that can lead to larger and fewer droplets. And first, there is like just merging via Brownian motion. So you know that a molecule, droplets, when they are in solution, they have Brownian motion, right? Which is like just random walks. And when two droplets, they kind of run into each other, they will merge. And merging leads to larger droplet. That's it. So that makes the droplet kind of larger. And theoretically, we can in fact write down equations to describe the size of the droplet as a function of time. So first thing to note is that the cube of the mean droplet size increases linearly with time, right? This is a linear relationship. And then second thing is then the prefactor, right? So the prefactor is big, which means the system will grow faster if you have like a large droplet volume, which means the droplets are kind of close to each other, so they can find each other faster, right? And the other is like if you have a small viscosity, which means the Brownian motion is fast, so then the system will kind of, the droplets will grow fast. Yeah, so it makes intuitive sense. And now the second mechanism is called the Oswald ripening, which is you do not really need to get in touch with each other in order to grow. You can in fact exchange materials indirectly through the dilute phase. So you can see like this, that small droplet, right there. With time, it just kind of gradually loses material to the larger one. And it's not in touch with the big one. It's just like mediated by the, yeah. The mean size, R cube, yeah. Well, it's mean size cube. It could be, yeah. You could think of it's the same. It's kind of the same. It could be like a slight difference, but it's almost. Why is this? So I don't know if I can derive this.
No, just intuitive.
Yeah, so-
One of the conserve water to go for 1/3. And if the length is typically the size of the drop, then maybe the length of the no, the length of the domain. So the domain would be the size of the drop, yes. And that will go time as to the power 1/3, which is
All right. But in fact, I can derive this because it in fact it's kind of easy to derive, which is like the number of droplets is going to change as kind of a square, right? Because it depends on the concentration. But then there is a prefactor. This prefactor is essentially the searching time, like how fast you can find each other. And the searching time has D in it, but also have like 4 pi dA, right? And the interesting point is that this D has KBT over the A, so that the A and A, this is a radius. Let me just call it R. The R and R kind of cancel with each other. Then you go this thing goes to like 1/n equal to a constant. And this 1/n is essentially like the total volume dividing by kind of mean volume. So yeah, so this is in fact not too.
That's like small first reaction, right?
Yes, you are right. Yes, you are right. Yeah, so this is not. Yeah, so you see like the derivation was done in 1979. Yeah, I always feel that because we are born too late to be able to have any opportunity to derive equations. Yeah, and so we were talking about this Oswald-Ripening case, and yeah, so for the Oswald-Ripening case, and I was just saying that like you have a small droplet, and now this time they do not even need to get in contact with each other. They can still, like the smaller one will still lose material to the big one. And the reason is because, the liquid droplets, they are subjected to surface tension. And so the surface tension increases the internal pressure of the droplet. And so specifically the pressure is like the pressure increase is 2 times the surface tension dividing by the radius. So different droplet size, like droplets of different size will then have different internal pressures. And the smaller droplet will have higher internal pressure. And because of the higher internal pressure, you also have kind of higher chemical potential. Then the molecules will like leave this small droplet and gradually go, there is a flow of the molecules to the dense phase. And so I was trying to, oh, this is very interesting. I don't know what's happened there, but I was trying to find a movie to kind of give a visual effect. And this is just an analogy. This is not exactly a droplet, but you could see like 2 bubbles of different sizes, right? When they are connected with each other through a tube, the smaller one does has kind of higher pressure, so it shrinks and goes to goes to the ears in the bubble goes to the larger one. All right. So okay. So let me. So now this derivation is even earlier. This is 1961. And essentially this the derivation of the tells us that droplet undergoing oswald ripening also has a linear time dependence for their average size cube. And now the difference is that the prefactor is different, right? Because we were talking about this is because of the pressure difference, and the pressure difference is because of the surface tension. So you could see the surface tension is definitely controlling the speed. And there is also terms like dilute phase concentration, because you really want the molecules to travel through the dilute phase. So higher dilute phase concentration, then the kind of faster the droplet grow. And also this diffusion now becomes the kind of the molecules, diffusion of the molecules in the dilute phase. Okay, so all these prefactors. Now, but you can see that in a realistic system, and one of the mechanism, right, either the merging or the oswald ripening will dominate. And well, which one dominate depends on the prefactors, but to the end, we are going to just expect a kind of a linear relationship. So how then does this really, if we measure this in the chromatin, if we observe the droplets in the chromatin, do we see the same behavior? So this is a question we started. And so this is the, as I was mentioning, that this work I'm going to present really comes from my postdoc work. And these are my collaborators. Daniel was a joint student between Cliff and Nat. And he's now a postdoc at Berkeley. And Cliff is the experimentalist. If you are in the condensate field, you definitely know him. And Nat was my postdoc advisor. Okay, so yeah, so now to understand how chromatin impacts droplet growth dynamics. So Daniel used a novel optogenetic system to create droplets in the nucleus. Okay, so now, so briefly, this system is that you have these red molecules, and these red molecules, they have intrinsically disordered tails. And these tails are known to have weak attractions between each other, but the attraction is not strong enough to drive the system to go through the phase separation on their own. And then the idea is that if you use light to kind of to trigger a conformation change in the green molecule, then upon this light, the activation, then these red molecules will bind to the green molecules. So now you form complexes. You have these hairball-like structures. And now you could see that each complex is decorated by multiple RDRs. And now the interactions between these complexes are strong enough to drive the phase separation. Oh, the light, this is it's called optogenetic kind of, it's a very high-end technique, which essentially I think it's the light will trigger a conformational change in the protein, which makes in principle, you have the red and green, and they are kind of complementary, but not exactly. And the light could make them kind of perfect matching so that then they bind to each other. Yeah, it's kind of a trick, or in fact, it's a very high technical science, but it's experimental side. Yeah, but this allows them to essentially manipulate the condensate formation using light. Or intrinsically disordered regions. Yeah, so these are just yeah, they are disordered regions of the protein, and they were essentially chosen from these IDPs, right? Kind of from these proteins that are known to phase separate. Yeah, okay. So right, so okay, so now so Daniel's really just shining light on the cell. And so these cells were expressing these molecules. And when you shine the light on them, the red and the blue bind to each other and the whole system goes through the phase separation. So Daniel then created these small molecules all over the nucleus. So these are the small droplets. These are all in the nucleus. And then these droplets, some of them merge with time and become larger. And so Daniel then tracked the droplet size and plotted the mean size of the droplet as a function of time. So this is what he gets. Now, surprisingly, this is way far away from linear. It's like 0.36 instead of 1. It's really like, it's kind of a surprise. But then we kind of, well, if you really like want to understand why, if you go back to like why these merging dynamics gives us, this thing gives us a linear relationship. That is because we were assuming that it's a diffusion, right? It's a diffusion process, and the diffusion, actually diffusion cancels with that radius here. But if the molecules are in the cell nucleus, you all heard about the talks. These are really not diffusive, but subdiffusive. Lots of things are subdiffusive. And when the motion becomes subdiffusive, this equation has to be modified. And once you modify this equation, you'll see that this exponent, in fact, is like related. So this exponent is related to this subdiffusive exponent. Now, so here, there was a scaling argument saying that, if you have MSD, which is T to the tau to the alpha, then the ripening speed should be T to the alpha over 3. So when you have normal diffusion, alpha is 1, you have 1/3. Sorry. It's like, if you have MSD, that is tau to the alpha, and then R cubed. It's essentially T to the alpha, yes. Yeah. So when alpha is 1, you get 1, and when alpha is smaller, you get smaller. Now, this is not exactly though, and I feel that there are, in fact, some theoretical work needed to work on it, but no one has. Yeah.
I was asked about that, because where you're deriving it, so basically
Yes.
you're modifying the 4-5 So
Yes.
how that comes about, you actually solve the diffusion equation with constant density and infinity, and then you have an absorbing particle and you compute flux of the boundary.
Okay, so
And
look,
that's how you just want. Like I said, it turns out that if you're in a high enough dimension, it asymptotes to a constant, which is 4-5. But
Mhm.
if you're slowing the diffusion down,
Mm.
so that it won't be as well mixed.
Mm.
Actually, remember what happens. So that coefficient picks up its own time dependency, but it doesn't go.
Coefficient.
So
you mean
it's a
if
much more
it's,
complicated equation.
you mean if this diffusion is not,
Yeah.
Oh, okay, yeah, so you're right. That is definitely another story I do not have time to talk about. In fact, many proteins, it has been found that proteins, when they phase separate, in general, like if you have oil molecules, you phase separate, you form kind of small droplets and they merge immediately when they encounter, right? But this is not true if it's protein condensate. In fact, there has been multiple studies showing that you can form 100 nanometer clusters, and since they are quite stable, they probably run into each other because they have to do Brownian motion in the solution, but they don't, right? So indeed, there are lots of things that should be modified here. And this one is not even a kind of strict derivation, it's a scaling argument. Okay. So yes, so this piece.
But I think you could make.
Yeah, indeed.
Is you solve now, not a diffusion equation, but a
Uh-huh.
solve diffusion equation.
Yes, you are
Not that
right.
coefficient, again, not-
Yeah, you are definitely right. But this work is really not my work. Okay, I put here for completeness, and also I did chat it with Daniel as my kind of really a paired friend on the So we chatted about this, but you know, most of the work is on his side, and he's an experimentalist. So you can see, there is a missing part on the theoretical side, I would say, and I don't know why I still haven't really found time to work it out. But subdiffusion is actually hard, right? You're so right. Yeah, but anyway, but subdiffusion is in fact quite hard, right? Like searching time for subdiffusion, especially these long-term memory type of subdiffusion. It's
if you're
yeah.
subdifusing, what happens? Your system is no longer well mixed. So all
Yes.
the assumptions you made.
Yes, you are right.
Everything is well mixed. Because that in the field theory, it all breaks.
Yes,
Everything slows down.
yes. That's why I didn't work it out, because it's a lot of messy things there as well, right? You might need to worry about initial condition or these kind of things. And anyway, okay. Yeah, you are right. Yeah, so it's kind of like this thing, right? All kind of forgetting even about that. Yeah, there are chemical, like it's kind of not chemical reaction, it's kind of like attachment, detachment, limited, right? Yeah, so all these we haven't really explored here. You know, the student went to the postdoc position and I went to so we all left the lab. And yeah, so this is kind of I would say kind of a starting point, but never has really been complete. There should be lots of work following this. But yes. Saturation. Yeah, well, so this one now, it really depends on, right, because chromatin is quite complicated and it really depends on how you, right, you have actually like more active region and more kind of less active region, right? And all these things we haven't really put into it. It really depends on like the motion of the droplet in order to see what kind of behavior will come out. Yeah, but that's, so okay, so but that's kind of one part, which is like trying to understand how chromatin impacts the motion of the droplet, which then lead to kind of modification of the merging dynamics. And so the next part I'm going to talk about is then like how chromatin impact the Oswald ripening. So Oswald ripening, you know, it's most significant when there are droplets of different sizes, especially when there are very different sizes of droplet, right? Because we were saying it's really the radius difference leads to a pressure difference. And so in that sense, now Daniel did this experiment, which he focused light on individual spots to create big droplets. You can see like there are 10 big droplets created in the system. And then he kind of just like turn on the light globally, but with a weaker amount, just illuminate the entire cell to generate the smaller droplets. Okay.
And the nucleus.
Yes, this is inside the nucleus. Now, I don't know if you can also find some in the cytoplasm. It depends on the protein. I guess the proteins are all imported into the nucleus. Yeah.
Scale.
Yeah. But this should be like something like that. Yeah. And yeah, so now you could see that each droplet, each large droplet is surrounded by many smaller droplets, right? So we were just talking about this Oswald ripening fact. And so we say that, okay, so then maybe because there is a very big internal pressure difference, we should expect the smaller ones to shrink and the larger ones to grow. And however, now Daniel waited for an hour, and it seems that these droplets, they are really not changing. Okay, yeah. So, well, we can further quantify the changes. Yeah.
Do you know how much mobility there is in enough? How much mobility there is in enough?
Mobility of the, yeah, so we know all these. We in fact tracked, no, even not us, before us, there are already like Cliff's group already has measured all these parameters, like diffusion of the droplet of the molecules in and out of the in and out, the surface tension, and the kind of each kind of volume of individual molecules. Everything is kind of being measured. And yeah, so which allows us, in fact, to quantitatively estimate what it should be if it's really like also a ripening dominated case. And it turns out that the estimation goes way faster than what is really changing with the actual droplet. And it's interesting that even this like first 1 and 2, it seems like 1 and 2 is increasing a little bit, right? But it turns out when we track back to the movie, it's really their growth is due to merging. Like the first one, the droplet one, you see a big thing on the left of the droplet one. It actually merges to one that causes it to grow a little bit. And that's it, yeah. So it seems there is really kind of no also ripening.
Which kind of state you
From which kind of state? So there is a cell, and there are like these proteins. They are kind of formed a vector and induced into this incorporated into the cell nucleus. So they express these proteins. They were there, but they were there, but they were in the like top form because the green do not bind to red. Yeah, and you have to shine the light to induce the phase separation. So kind of like really suddenly you kind of tune the system into the super saturated situation, and so it depends on how you shine your light. You can create different patterns. Yeah, essentially that's what we see, and that's which is quite different from the theoretical expectation. So again, now there are multiple, yes. Growth at the beginning, why there is a oh, no problem. You mean there? Oh, let's see. I don't think it's a jump, but it's probably because Daniel, like, these are short-term measurement and these are long-term measurement, and probably in different cells, and it's really cell heterogeneity makes, yeah, it's kind of also like here, your substitution, you see the short-term and the long-term, they actually have a shift. It's, yeah, cells are messy. Yeah, so it's kind of yeah. Yeah, so these are, yeah, so we do modeling, and yeah, so I wanted to say that there are actually multiple explanations for this kind of situation. It could be like, as you were already kind of talking about, it could be attachment, detachment is slow, or many other things. And we tried to make up a model saying that it's the chromatin, and it's the elasticity of the chromatin that's really limiting or slowing down the oswald ripening of the system. Now, whereas I would say it's probably true that the elasticity of the chromatin is going to contribute, but there could be other factors as well. So we are really just highlighting factor. I do not think that this is a whole story, but still, as a series, there are limited things that we can do unless we have experimental collaborators, which I will be super happy to collaborate if any of you are interested. Yeah, so this is what we do. We essentially come up with the simplest simulation that we can do, which is to first simulate molecules, space-separating molecules as beads, just spherical beads. And then we simulate the chromatin as chain, like chain of beads, kind of be the spring chains. And then to make it known that chromatin is quite, well, chromatin is for elastic, but what elastic side depends on different cell type though. Yeah, so we added the cross-links to cross-linking different regions of chromatin. And then we make attractions between these spheres, these particles, the red particles, just to mimic this, like their phase separation ability. And we also make these red molecules kind of repel the chromatin, because that is what is observed, that like there are holes in the chromatin where the condensates are. Yeah, so you can see red attract each other, and all the rest of the molecules, they are kind of essentially like short range repulsion. And then we have also a chain connected for the chromatin and some crossings. That's all. And we evolve. Yeah. Okay. Sure.
Just a question to understand better the model. So you said that your chromatin model is like a polymer network.
Yeah. Yes. Yes.
Crossings are permanent.
Yes. Right now it's permanent. Yes. I hope we can make it reversible. Yes.
So condensate they attractively interact with each other and also with the chromatin. Or not?
With chromatin, they do not attract
So
chromatin.
just
Yeah. So right now it's really just repulsive
So
kind of.
just want to check the impact of, let's say, chromatin the substrate. How it
Yes.
affects the
Yes.
formation.
Yes. 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 Okay. So then we evolve the whole system through long-term dynamics, which is essentially just a kind of to equilibrate the system. And so here these are the snapshots in the simulation. You could see like these droplets they are coarsening in a cross-linked chromatin network. And you could see that this initially we have these red particles, these red molecules, they are kind of distributed randomly in the chromatin network. And as we kind of tune on the attraction between the red molecules, you started to see these kind of small droplets forming almost everywhere, right, all over the place. And then with time, they started to merge, some of them coarsen, and to the end, we see two droplets in the simulation box.
We keep that going, you get one.
Yeah, so that's the question, right? So yes, so we now, in order to know whether if we keep going, it's one or two, we track the size, essentially size of the droplet or number of particles in each droplet. And you could see like many of the droplets, smaller ones, they die at a certain time and only leaving the two, which seems to be quite flat here. So they could kind of coexist for quite a while if this trend continue to, right? And we actually found another interesting case, which is this one, which is that you could see that the two droplets now, the blue one initially is larger than the yellow one. However, with time, this larger droplet gradually loses material to the smaller droplet, and the yellow one started to grow. And this is kind of like reverse of this Oswald ripening effect. So this, yeah.
Someone depends how much of your polymer you have there and how much of you compensate particles, right? Because if there is a lot of polymer, there will be no room to build them.
A big one. What is the
Yes, yes, yes. Yes, okay. Mm,
for the chromatin polymer?
mm.
It will be the one that.
So we have this 7 micromolar. I don't know if it make any sense. Yeah. Yeah. I
Some where you thought not for your system.
mm, mm, mm. Yeah, so all is indeed, it's kind of embedded in the cross-linked chromatin network, right? If you have more cross links, now you have kind of more droplets of smaller sizes, and they can all kind of, because they grow and they kind of stop at a certain stage.
Without changing the density of your top, the dominant.
No, just adding more cross link. Yeah.
Could you explain what's going on in that middle one, where the yellow one grows at the expense of the larger blue one?
And it could be that like the blue one is unlucky and grows in an unfavorable location. It's like as time goes on, it feels more and more restricted by the surrounding chromatin. And there is another one, the yellow one is in a happier place. And
Go into your simulation.
yeah, we could, yeah, so you are right. Okay, so you are already talking about my next slide. Yeah, okay, but oh, and I'm already an hour. Am I already an hour? No, not yet. Okay, great. Okay, yeah, so we also kind of remove the cross link, and when we remove the cross link, we see more merging impact. So it's interesting that the chromatin, like the cross link, is really kind of slowing down the merging. It's like really compartmentalize the droplets, and they are not. Yeah, and when we remove the totally the chromatin, the ripening suddenly becomes very fast. You could see like the time, you could see the time scale is 10 times faster once we remove the chromatin. Yeah, and so, okay, so here are just like more kind of, for each simulation, we now do multiple times and find the mean behaviors, and you could see that when you have no chromatin or no cross link, these kind of ripening or the merging dynamics essentially is a linear relationship, as we were kind of expecting before. And as we add a cross link and as we add more cross link, it becomes kind of progressively curving down, right? So that is, it becomes kind of anomalous. So yeah, so okay, so let's now move to the slide you're proposing, the idea of proposing. Yeah, so first we see that, you know, there are stretched chromatin around this droplet, and we could really just find it in the simulation, because simulation is everything in the simulation, everything is clear. You can track down all the like bound lengths between the chromatins, and you could see that they are stretched. And so, and the other question that we ask is, so where the droplets, like where the droplets form, do they form randomly? So when we see this thing, when we see the droplet, are they just in random locations, or they are really in the like kind of formed according to the local mechanical environment? And to see that, we essentially removed our droplet and insert a sphere, and we can measure the pressure from the chromatin on this droplet, on this kind of sphere. And now we found that this pressure on this sphere where there is a droplet, the pressure is low. And if we just insert this sphere any other places, the pressure is much higher. So indeed, these kind of the droplet, they can sense the environment, and they are kind of almost guaranteed to find a nice position to sit. Yeah, so that is another finding. Yeah, so finally we did some kind of small theory. I wouldn't call it a theory because it's really just some quantification. It's like quantification of experiment. It's simulation is our experiment in a way. Yeah, so we found that, so first we measured so first we can really just measure the pressure on this sphere. And what we found is that if we remove the crosslink, the pressure, it looks like the blue one. Okay. And I don't know if this looks weird to you. It should be kind of a flat line, right? If you have like no crosslink, you should kind of expect a flat line. So this curving up thing is actually from some polymer physics, but we are now going to talk about that. Essentially, the difference is clear, right? When you have crosslink, you could see that this is called a strand stiffening, right? When you have strand, in this, when the size grows, you progressively kind of stretch the chromatin network, and which makes kind of the pressure from the chromatin to the sphere becomes larger, right? So this is the difference. And so this stretching of the chromatin and the feedback of that stretching to the pressure depends on the local location, right? So we put this, now we put this kappa here as kind of which has a location dependence. And so we just write down the equilibrium conditions for the droplets. It's like really just thermodynamics. You have temperature, you have chemical potential, you have pressure. They all have to be in balance. And then you know, then we can write down like what's the chemical potential in any location in the kind of cross-linked system. So, and we also have a number of particles conserved. And essentially what we come up is this view, which is, you know, there are softer regions which correspond to the most right part. You have kind of a chemical potential which comes down and goes up, but it's kind of lower. And if you have a very stiff region, you know, the chemical potential is very high, and so that you have almost no chance to form droplet. Okay, so I guess, you know, the physical picture is very clear, right? You have just softer regions and the droplet will grow preferably, and stiffer region is kind of less. Yeah.
So these are pressure, so this is of let's say pressure, because of the chromatin of the
Yes.
But
Yes.
Your crossings are randomly distributed, I guess. I don't know.
Yes.
Why is there are different regions of pressure?
Oh, just a random fluctuation maybe. So I haven't really even model like heterochromatin region versus euchromatin. That would be even more drastic. We really just throw in random crosslinks, but due to just the stochastic fluctuation or noise, right? You can just randomly, you just kind of by luck having more in this region and having less. Yeah. By chance, that's it.
Your do again similar simulation. Will it sit in the same place?
Yeah, that's a very good question. Yeah, I'm asked multiple times for this question. We found that almost 50% of time they'll be in the same location. Not always. And I guess maybe there are some degeneracy in the cross-linked network, right? Maybe this location is comparable to that location. So then the system could prefer another location, right? Because, right, do you see that? But indeed, we see like 50% of time, we see the droplet in the original place. Yeah, okay, so, and we were just drawing the analogy that, you know, this really looks like you just have different potentials in your, in the local location, right? And you could see water, the water petals form on an uneven surface. This is what happens. And yeah. Yeah.
So this kappa,
Uh-huh.
that's the what is it?
Yes, it's kind of not bending modulus, but what's that? It's like when you expand it, it's not that you bend it, but when you expand it, you have this elasticity. What's a Bach modulus? It's but essentially it depends on, it's the slope when you grow, like the pressure increase versus this radius. That's the slope of the relationship. It's like as the droplet grow further, you have more and more pressure, and that is the slope. It's it's which I measure in my simulation. Yeah, it could be better if you can measure it in the experiment.
And the temperature, dense space, and the dilute phase, they should be the same. So
Yes.
what does that condition mean? That condition does not. Do you use that condition?
Yeah. I used the temperature and the chemical potential balance in the, so the chemical potential, it's like if you were to put a droplet here, the chemical potential in the dense space will be here.
Yes.
So, but the
potential, I understand,
yeah.
But this is the canonical ensemble, right? So the temperatures should be the same.
Yes.
I don't understand why does a separate condition that is required for.
Oh, you mean I have 3 conditions? I really only need 2.
That's what I.
Okay, yeah, you are right. Yeah. So in fact, I only need 2 because you are right, because say pressure is in fact a function of temperature and the chemical potential. So we really need 2. I write it in this way is because like using this pressure relationship, it actually gives us a chemical potential relationship. So I spelled it out, but you are right, because now inside the droplet, because we only have a single component, then 2 parameters are enough to describe it. Yeah, if we have a mixture of other components, then maybe we need more condition, but indeed you are right about this. Yes. Okay, so this is yes.
It's an experiment.
Uh-huh.
So if you imagine, so in this case, these proteins are not interacting with the DNA. They're creating this region where they self-interact. And so in a biological system, that would be something like nucleus, like you pointed out.
Yes.
Self-interacting with RNA. But that means that if the chromatin stick like crossing chain, you see a lot of crossing as well.
Yes. Yeah.
What that is just from the cross-linking chain, your nucleoli will either merge or not. Based, like, because it's different cell types. You see different
So you are totally right. Now this model is really kind of a beginning step. It would be better if we can use reversible cross links, right? So that like right now, because the cross links are permanent, so we can only really look into the impact of the chromatin to the droplet. We are only seeing how chromatin impacts the droplet, but we are not really seeing how the droplet can impact the chromatin, right? But in realistic, like real cell, they are kind of, it's really interactive. The growing your droplet could break your cross-link, right? And then the formation of cross-link could constrain your droplet. So it's an interplay.
But maybe the reason why different cells have different numbers of nucleoli is because
Could be possible. Yes, it also depends on how fast. Now, first you have to think about when the nucleate, how many nucleated sites are there. Then it's about how fast they can move, or probably not ripen. Can they ripen? If you ripen, then you lose the nucleolus, right?
So in the
Yeah.
cell show, there's this huge hole, right? But this is great. The cell very dynamic. It's very prosthetic.
There definitely, yeah, there are cells with multiple, right?
Yeah. But as soon as you differentiate these, you see the big, you see many. That never come.
Okay, so I haven't thought about it. I was most just like, I know that there are cells which has only one, there are cells which has multiple, but I haven't linked it to the reasons you were saying, right? You might be right. You might be right. Yeah.
Sounds like someone.
Yeah. Yeah.
If
You can't imagine.
yeah.
Maybe someone. Okay. Yeah. Yeah, okay, so I'm really at the end of my talk, and I guess the only thing I can talk about, like sulfate, might be that say, could there be like some feedback loops, which is saying, say, if you have transcription condensate, right, it kind of like repels the chromatin and makes the local kind of environment more porous, right? And then that could allow the transcription factors to come easier, and then you are easier to form this condensate. It's kind of a memory effect. Would there be such kind of thing? That would be the only.
Push that in. The RNA would push away, and RNA. RNA is not going to
I see.
When the RNA is gone, you bring it back.
So yeah, so it's really like about the dynamics and how fast. So I feel that I'm not having a very good sense about, say, how fast all the things coming back. The other, yeah.
Well, the RNA be made by the DNA, but when the RNA comes, it's not. So at that moment, you get a transient.
So it's the RNA that.
Once the RNA is finished being made,
then they'll go away. Yeah. But so it's like this kind of process, would it really have an effect? It's really about like how long you can remember that you have formed this RNA bulky thing, which kind of opens up a whole. But these are all like.
The polycomb will bind the RNA.
Oh yeah.
The polycomb will bind the RNA. That could create space. Create space. The polycomb.
Yeah.
Okay. Well, yeah. So I really want to say our chromatin model is like really like the simplest model we can come up. But you have like there are in the field way kind of sophisticated models. But the model really needed to be incorporated is a dynamical model, which not only help you to create the snapshots, but also can produce kind of right dynamics like viscoelastic, not just the elastic part, but also viscous part. That I feel very hard to really to work it out. Yeah, and I'm not really, I'm kind of not really in the chromatin field. I see like you use high C data to create these chromatin models. Are they dynamically also correct?
This is why we start. Well, it's like you're gonna see a contact, but it could be. Yeah, so you can see this, right?
Mm,
Because you don't know how long they maintain, you do the simple
Mhm.
science, get the snapshot, right? And you see that, but you don't know if it's there 10 minutes later. It could fall apart.
mm, 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 mm.
Yeah.
Yes, sure. Yeah, I'm done. Thank you.
I'm gonna ask a basic question,
Mm.
very beginning.
Mm, 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 mm,
You were showing us
mm,
under elimination of light,
mm, mm,
was
mm.
it just being similar in any perspective?
Mm.
Because generally, the cell within itself is not
Uh-huh.
light.
Uh-huh. Uh-huh. Uh-huh.
What is
Why?
right?
Uh-huh.
The reason why itself, and what is its L? When the condensates are very large, when they disperse them.
So I guess like Cliff created this tool really to be able to precisely control the number and the size of the droplet. It's like for better control. So his, at least a part of his interest is in soft matter side, right? This kind of becomes a more, yes.
What is that?
Uh-huh.
Right?
Yes. Mm-hmm, mm-hmm. Mm-hmm, mm-hmm, mm-hmm. Yeah, so there are endogenous condensates as well in the cell, and there are different size distributions, I believe. It's like, it depends on, do you kind of, because optogenetics, it's kind of, it's kind of you're sending the protein all at once, right? But in the cell, you actually kind of gradually produce proteins and kind of gradually added your proteins into individual droplets. So that is kind of a different initial condition compared to the optogenetic system, right? Yeah, so in that sense, this could lead to like different droplet size distribution or, yeah. But this one is kind of easier to study once you have this kind of control. Yeah.
Yeah. But or more, bit more detail about your Chromatin model. So, again, you said that you mentioned that 7 micromolar.
Yes,
So,
yes,
but that
yes.
is,
I understand. So I would say like, essentially for this droplet, which is about 1 mic, less than, I guess 0.5 micrometer in radius, this droplet. Okay, so the simulation box is like 2 micrometer, and then the droplet is kind of half of that in diameter. And so for this droplet, the cross links are almost all stretched at this. Yeah.
But just talking about the polymer part.
Uh-huh.
So how many did the estimation about
Okay, so 7 micromolar, let me think. 1 nanomolar means 1 like 1 nanomolar is like E. coli protein. It's like 1 protein E. coli, which is like this. Right. So we have 7 million micromolar, which means like at least the length scale will be 100 nanometer length scale. Right. So it's like, oh, because this is like 1,000 times this one, so the volume should be 1,000 times smaller, so the length should be 10 times shorter. So it's kind of every 100 nanometer you have a cross link.
Okay.
Of course, the chain is not stretched. The chain is like this.
Sure.
Right. So then you can stretch the whole thing until a droplet. You form a droplet, and now this chain is stretched here. You have crossing here.
And the polymer densities.
The polymer density is very high. I would say probably 20% or even higher volume fraction.
Okay, no, that's not.
Yeah.
20%
Yeah.
And
Yeah.
okay, and So you have, okay. Okay. And,
So look, simulation to the end is what you put in, you'll get what it comes out, right?
No,
So
okay.
yeah, so in that sense, only if we can have like right numbers from experiment to put in, then we'll have these are okay number. These are okay numbers, I know, but
Yeah, I quite a few version behavior.
no.
well,
These numbers. No, it was just-
well, I would say all item simulations have the ability to really show you something you can't think of. But these simulations, well, it also depends on how hard you think about that, right?
Different, instead of having cross things,
Mm.
you have just entanglement. So whatever
Ah,
you said,
entanglement,
you
I
have
see.
only this,
Uh-huh,
because if your chain
Uh-huh.
is now,
uh-huh, uh-huh,
this should be
uh,
roughly
I see.
cross,
uh-huh,
provided
uh-huh.
you have made the
Uh-huh.
mineral, everything your condensates are
Mm.
smaller than
Mm.
gravitational time.
So yeah, so I understand that entanglement should be important. The problem I would say is that these simulations seems to entangle. So okay, so repetition is slow, but if you have a droplet which grows really actively apply pressure, then even if you have entanglement, it can be it's kind of like can be pushed away.
No, sure. But, I don't know, as far so if let's say all the times here that are relevant to I mean, I don't know, I guess it's shorter, they are shorter than the entanglement time or the repetition time of the chains. Probably you should be see much of it, or maybe it, I don't know what you said from the pressure. I guess cross-link, I mean, use cross-link.
Yeah, cross-link, you kind of you use strong enough cross-link, then even if you have pressure, you can push it.
No,
And
sure.
yeah, entanglement, it depends on how strong is the entanglement. Then yes, volume fraction matters and initial kind of, yeah. You mean 20% is the yeah, we in fact have, yeah, our chains are very long, very long. Yeah,
So
yeah.
maybe see, or maybe you see something.
It could be some of the effect is actually from the entanglement. That is possible. Yeah, that is, yeah. But when we remove all the cross links, we do not see this. So
because how long are your chain? So it is like a mental differential of different length. Like
I think I have 8 chains.
8.
8. Yeah.
And how long? How many more?
They are super long. Let me see, 10 to the, it's kind of 10 to the 4 beads.
Can you equilibrate them first?
I don't know how to. I'm not sure if I have equilibrated them. I have tried to equilibrate them. Yeah. So it could. Yeah. We do spend quite a long time to just wait for equilibration, but I can't guarantee if it actually equilibrates.
No, sorry for this.
No, we can chat more. Yeah, yeah, we can chat more.